So today we are starting to share our research with our peers. Our intention for doing this one basically is like kind of knowing what others do, then you can in future share what, like, think about a project that you can share each other, or think about the area that you can integrate your research work with someone else. So before I start, I wanted to say that Alexis from the Southern Illinois University, she will be not like in person today because her flight schedule changed, but I have recorded presentation on behalf of her. I will share with during her presentation time. So the presentation order will be same as what I noted in the agenda. Before I go with the presentation, I have few updates from what we are doing in the BPRI. So first of all,
well, we want that separately. If you're trying to natural or like,
what's the sound? Margaret, can you please? I think it's your mic.
Oh, sorry. I thought it was off.
Okay, it's fine. This is just us, so no need to worry. So go to this Slido.com. I don't like just, I have 2 polls to get the date for the next meeting. That's why I created it. Another thing is, I was thinking if the presentation run longer time, how can I get the all QIA question and answer? Then I have some kind of record for the future discussion, especially for the Alexis presentation. If you have questions, please post in here. Then we can get back to those questions during the next meeting when she is in the presentation. Okay, so this is, I'm also new to this tool. So if you know someone know more about Slido, please share if I'm doing something wrong. Okay, so as always, I will highly encourage you to acknowledge whatever other people sharing, and also note that this is a interdisciplinary team. Some may not understand your technical jargon because they are in different field. So when you are presenting your presentation, try to use very simple kind of conversation style words, then everybody can understand, especially kind of me, who are kind of apart from you whole research. So I hope I can understand a little bit. So the next one is what we are doing all throughout this whole month. So we have to first say that from the SIUE team, Dr. Rick have kind of initiated distributing our research engagement series. What we are trying to do is highlight research activities in each BPRI lab. We start from the Southern Illinois University. Dr. Brittany have volunteered and share her research through the research engagement series. If you didn't register to the GLI network, please register and get update on that. In additionally, while we posting research engagement series in GLI network, we notice that we can promote BPRI work through social media. Thanks to Vivian and Odelia's help, we are posting, cross-posting all the things in these 2 social media websites. Especially they will reach out to you and ask about like 1 or 2 sentences to talk about your research. Then they can post something related to your research. Okay, so that is very good opportunity for social media presence for you as a researcher, because if you start earlier, your visibility will increase when you're applying for a faculty position. Okay, that is really important. You will realize that when you prepare in faculty package, they asking a lot. So you need to prepare in advance for that. This is one tool. And next one is few updates on our overall process of the BPRI. So now we are moving into the second stage of BPRI. So we all identified that our trainees can engage in any committees in the BPRI as a volunteer. That is, you don't need to participate every meeting, but if you have time, please join one of these committees and get a sense of what is the project administrator is look like, especially if you are a PhD candidate planning for the academic job. You need to learn about how project management works. So you can join these meetings and get kind of sense of feeling how you have to prepare yourself. Especially I invited Vivian and Odelia for outreach committee. I think they find it very useful. Vivian, you can share your feedback in sometimes during this meeting. So if you are wanted to engage in any committee, please let me know. Then I will share the meeting details and when they are happening, then you can join it. And also we are drafting our second strategic plan. If you have any feedback that you think as a BPRI want to improve, please let us know. Okay, so that is kind of because you are one core part of our project. So now let's move into the presentation. So I'm sorry, Vivian, you have to go first. So I will stop sharing. And if you have a presentation, you can share. But if you like to talk with like a casual presentation, please let me know in advance, like before you starting. Thank you. Oh, we cannot hear you.
Can you guys hear me now? Okay. I was just gonna casually speak. I don't have anything prepared. So I'm still kind of in the beginning stages of putting my plan together. But basically, what I would like to study is the maternal effect between moms, female locus of S. gregaria and baby hatchlings. And basically, a lot of research has been, not a lot, I guess a good amount of research have been done before, and it's a little bit old. I think they stopped working on this about in 2008. And a lot of things are still unknown, and I kind of want to pick up on that. Also, for example, there's a chemical that female locusts deposit in egg foam of babies, and the babies get coated with it, and this is believed to determine the face status between solitarius or gregarious of the hatchling. But I think the last paper that came out was in 2008. They believe it to be L-DOPA, but they couldn't further test it because at that point, I believe the researchers were in Australia and they could not import S. gregaria. So I kind of want to do some experiments with that. I'm interested in looking timing of crowding, what effect that has on the reproductive tract cells of the mom. There's actually a lot of things I want to look at. There's also outside of the other experiments of L-DOPA, I would like to do some maybe look at the RNA of developing embryos for the eggs. So I'm still kind of putting everything together, but basically, I think my overall idea is that I hope to understand how and the pathway of how the mom is helping the baby choose not baby choose, but determining the face status of the baby. And yeah, so that's kind of what I'm putting together. I believe that this is also like a really cool example of epigenetics. And yeah, that's kind of what I have together so far. I'm still kind of putting the whole draft together, but yeah. Any questions, any comments, any ideas?
Thank you, Vivian. So because we need to I need to reiterate one thing, because we have some trainees who starting PhD like just soon after undergrad, and some trainees are with masters. Okay, with masters, they have kind of broader knowledge on how research preparation, but with who are starting with bachelor's, they are kind of still figuring out the things. So when you commenting, so when you're thinking about, oh, she don't have some progress or something like that, don't think like that, because they're still in the PhD journey, starting this to realize what it look like. So especially if you're a senior PhD candidate, if you have any idea to share with Vivian, I'm opening the floor to you. So please share it, like you can share with research in respect, and also anything that you find helpful when you preparing research ideas as a student. Generally, you can also share with Vivian, because she may appreciate that kind of advice. Anyone?
Are we speaking or are we putting it in the slide?
You can do both, but mostly Q&A session is for the Alexis presentation, because she's not here.
Oh, okay, okay.
So for the others, you can share your feedback directly.
Even if you're interested, I was actually reading this paper since you mentioned that you're talking the maternal effects on moms and babies. There is this really nice paper. You may have already read it, but I was just sorry if it has not crossed. It is parental phase status affects the cold hardiness of progeny eggs in locus. It's published in Functional Ecology. It's the leading author is Wang. So if you want, I mean, I can send it in the thing that Tanya has shared. In this way, you don't have to like read the, write down the whole thing. But I really like the paper, and I think it really overlaps well with what you are interested in studying. So if you are interested, I can share that.
Yes, please. Thank you so
Okay.
much.
Yep.
So any other comments? Okay, one comment from me, Vivian. This is general comment that what I found in my PhD journey. I also had a lot of kind of ideas that I wanted to do with my PhD, but at the end of my research proposal, I kind of like, I had a whole fruit basket. I end up with one grape kind of thing. So when you are like trying to prioritize the research topic, when you're reading the literature, try to identify that everybody say this is the gap and focus on that, because that help you to publish, because ultimate your goal is publishing. So just kind of my feedback. So as we don't have any comment, I think so far, no in here chat. So let's go with the list. Lisa.
Hi, can you guys hear me?
Yes. Do you have presentation or you'd like to talk?
Yeah, I have like a little bit of like 3 slides.
Okay. I think you can share, let me know.
Yeah, I can, I think. Can you guys see it? Can you guys see the presentation, right? Hello? Yes, now, okay, because I couldn't hear nobody. Thank you. All right, so our lab is mainly focused on like studying the locus olfaction and like from the point of the brain. So what's going on in the brain when locus sniffs something. And me personally, I'm a first year PhD, so I'm same way figuring out stuff for now and just putting all together like the ideas that I have that I might study further, might not. But to just give an overview, what we do in the lab is, for example, so we study locus olfaction, and I personally study it from the point of the electrophysiology. So I do the electrophys on locus brains, and I look for like the response with electrophysiological activity to certain orders. So here you can see on the slide an example of locus surgery. So it's like a prep that we do every time we want to run any study. So first we like mount the locus on this plastic platform. So like we kind of like put its neck in the right position, so we fix it. Then we build a wax cup around it so it can hold a saline. Then we open up the brain and we put it on the platform so it's like easier to reach. And then here you can see the different areas of the brain. Me personally, I would like to focus more on like antenna lobe. And for now, I'm focusing on projection neurons. So what I do, I don't know if you guys are familiar with like patch clamp technique. I don't know, give me like thumbs up, thumbs down if you're familiar or not with it. Okay, cool. So like people know what patch clamp is. So I do a patch clamp. And right now I'm trying to figure out and build the experiment that would show if there is any connection between serotonin release in the brain and olfactory response, and then compare it between the Americana locus that we have in our lab, and we don't have Gregaria colony, and then in the future, hopefully compare it to the Gregaria in the solitary stage and gregarious. And here is just like an example of like the response we can get to certain orders. So this is an example of the on response. So meaning when the order is presented for 4 seconds, which is, you can see it in the blue bar, you can see the increase in spiking. And this is an example of patch clamp on the projection neuron. So it's the neuron that actually has a spiking activity and it fires actual potentials. We also have a off response, but I don't have data yet collected for that. And also to see if there's any serotonin even within the locus brain. I'm currently developing a protocol to do in immunohistochemistry on locus brains, which has been a little complicated because not a lot of people have done the imaging on locus brains. And every protocol I find is completely different. And out of all protocols, I have only one that worked somewhat, but I messed up a little bit with mounting the brain, so I couldn't capture a good picture of it. But hopefully within 2 weeks, I will have a really good pictures. So I was staying for serotonin. And when I do patch clamp, there's a technique where you can like fill the cell with neurobiotin, which is basically like a tracer where you like, when you patch into the cell and then in the end, you apply positive current and you let the neurobiotin like swim in the like cell body, and all the dendrites, they go through the brain. So the ideal experiment that I'm trying to have is, so I would patch onto it's for now, like it's like a starting point. I would want to patch into the cell, fill it with the neurobiotin, and then stain it for serotonin and see how the difference in response to certain orders can be or cannot be regulated with like serotonin. So like we know that there are different types of neurons and cell bodies within the locus antenna lobe. So for right now, I'm trying to see if serotonin has any effect on the firing rate and response to certain orders. So I have one, it's not working. So this is like sorry, computer being slow. So this is like one of the example I did on like staining for serotonin, but I know that this picture is not like great at all and has a lot of like unspecific staining, but that's just like something that I have. And then the future goals for the experiment is to perfect the immunohistochemistry protocol for now. and then continue the electrophysiological experiments, filling the cell, as I explained before, staying for serotonin, and observe if there's any connection between serotonin presence and in the cell and the auto-evoked response. So that's it, and I'm ready for questions if you have any. I know you might have a lot.
I couple questions there that wasn't completely clear to me. So you're patch clamping the soma of the projection neurons then?
Yeah, so cell bodies.
Okay. Have you tried doing sharp recordings of the PN neurons, or is that not possible?
Say it again, I'm sorry.
Sharp electrode is instead of patch.
No, actually, I never done the sharp electrode recording, only patch clamp. I mean, also like in the future, I'm still like, you know, I'm trying to like set up first experiment. Then if we get something, I will move to like next. We also have the extracellular rig, which probably will have like a bigger picture, like because you can record from multiple cells at the time. So that's like, if there is something, I might look into like extracellular, but no, I never done the sharp electrode recording.
Okay.
To answer your question.
Yep, just because the soma is kind of electrically distended from the cells, so you can pick up the action potentials, but very minimal kind of subthreshold activity. Where you could with a sharp. The other thing I was going to say is, if you haven't already spoken with Herman, you should speak with Herman about serotonin staining.
Oh, no, I think Dr. Rahman mentioned speaking with them because so now I think I have a protocol that is working good. So he was like, yeah, let's try this last protocol. And if nothing works, we like go out there and like talk to people. So I'm like,
because we did, so Hongsha, Herman, Fabrizio, and I had a paper from last year on antibody standing in locust brains, but we were actually using the serotonin antibody as our test. So to make sure that everything worked, we were doing serotonin stains in the locust brain. So to get that working, we had to do much of that debugging. But yeah, so if you are still working on that protocol, Herman has done a lot of 5-HT stains and probably can help you out there.
yeah, I would love to have like to talk to that person or to have any protocols, because for now, like I started with like NC82 just to like have like at least a starting point of like get something right that's working. So NC82 works. So now I'm moving on to like staining for serotonin. So I think, yes, I need somebody who has expertise in serotonin work. Thank you.
Okay. Yep. You do, I assume you've met Herman. He's a professor at the BPRI.
Say it again.
Dr. Dirac, he's a professor with the BPRI at Baylor.
Yeah, actually, Dr. Rahman mentioned somebody at Baylor who does work on serotonin.
Okay. Yeah.
Yeah.
So, yeah.
Okay, Liz, I will take the kind of lead and make sure that share the details of Dr. Herman with you. Then you can contact him directly.
Thank you.
Oh, any other questions for someone? Is there any other questions? Thank you, Liz. I think everybody will be happy with your presentation. Let's go with the Jiayi. I think I pronounce it correctly.
Yeah. Can you hear me?
Yes, we can hear you.
Hello, my name is Jiayi. I'm from Jackson's lab in Baylor College of Medicine. So our lab is the sequencing lab. We do single cell genomics as well as transcriptomics. And my project here at BPRI is a collaboration project with Professor Lab. So their lab, one of their aspects is the LGMD cells. So these are locust giant movement detector neurons. So these cells are in the locust brain that is important for collision emission as well as detecting these giant movements. So these cells are big and they are important neurons and they have a robust response to the signal inputs. So these have been really nice models for single neuron modeling. And while these cells have been studied in the biophysics aspect for decades, we actually do not know much about their genetics or transcriptomics. So our lab has developed these single cell sequencing methods called MapQC, and I would like to share one slide to show hold on, let me. Can you see my screen? Yeah, this is just to give you an idea of how this method works. So this is the MapQC that our lab developed. So it's a total RNA sequencing method to detect the transcriptomes in single cells. So on the top here, we have the your mature RNAs or immature RNAs doesn't really, we can detect both. We use a G18 enriched primer as well as use the detail to capture the mature RNA or not to just capture the inner sequence of the RNA molecules. And we will do a reverse transcription where you will have your RNA DNA complex. And then we do a 10 rounds of modified annealing to have efficient priming of the RNAs. And then we digest the original RNA template, do a poly-C tailing to add in the C bases for our newly synthesized fragments. And then we use a 3G leading primer to do the second strand synthesis so at this step we have the double strand DNA molecules that is ready to be amplified in PCR and go ahead to see. Just a general idea of how these methods works. And our method has improved coverage. We have less bias on the ends compared to a previous method that's total RNA based. And also we have better sensitivity in these lowly expressed genes. And our and we based on the ERCT specking. So this is just a method to detect the sensitivity of single cell RNA-seq, just a kind of a standard assay, and we can reach 90% detection of a single RNA transcript in a given cell. So my project is just to since Professor Lab is able to just dissect these single LGMD cells from a locust. So we are able to collect it in a tube and do the single cell sequencing to see what genes is expressed in the cell in the cells, and we can compare it with the brain bog of the locust to see what are special of these cells compared to other types of neuron or brain cells, to see what may be important in their function. And we have pretty much finished with the sample collection, and we'll go ahead to sequencing in the following month. So that's kind of that part. And I'm happy to answer any questions, though I don't know much about the biological side of the LGMD and locust.
So anyone have questions? I know that for the most of our project team, the single cell, the Trainees is so new. I remember faculty members also telling in the meeting, we need to go to the Chuck's course to learn about that. That's maybe the reason they are silent. So don't think that you did a bad job. You did a good job. everybody is kind of new to this technology. Anyone have any questions? No? Okay, let's go to the Mehreen, I think.
Yeah, can share my screen. Can you guys see my screen? Okay. Okay. Hi, everyone. I am Mehreen here, and I am a first-year PhD student at Arizona State University, directly advised by Dr. Arian Cease and co-advised by Dr. John Harrison. And I'm actually the part of the Global Locus Initiative Lab. I know Tanya mentioned in the beginning that if you have not joined the GLI forum, it would be a good time for you to see. We have our Twitter handle as well as our website if you would like to follow us to stay updated on what is going on in our lab and what has happened in the past, so you can do that. We are very excited to be on the BPRI team and collaborate with an amazing team as you all are. The overarching question, I mean, that all of us as the BPRI committees, the researchers are trying to study here is what responses are conserved across species and environments, and why and how is the major question that we are trying to answer. For ASU, we are actually leading R9, that is integrating lab to field research. And in our case, specifically our research interest as of now, and also as more of this project develops, we want to study the phase dependency on the nutritional physiology in Cysticerca gregaria. So what we want to study here is that how phase change, such as if the locus is exhibiting a sartorius phase versus a gregarious phase, then how does that phase change affects what they want to eat, or does it have any effect on what they want to eat? Primarily in respect to, I know carb and protein studies have been done quite a bit by a lot of researchers, and the literature is actually present for those studies. But we are also interested in studying carbs and lipids. And why we're interested in studying carbs and lipids, that is, reason for that is because we are trying to understand the diet to the migration. So to make it simpler, make it much simpler to follow, I have created here this flowchart that explains how does the phase change affects the plant-insect interaction, and why does the phase change affect plant-insect interaction. So to answer the how question, how it affects the plant-insect interaction, we are trying to understand whether the sartorius phase or the gregarious phase of Cysticerca gregaria has a host plant choice, such as are they going to be, if you give them a carb versus a lipid diet, are they going to be going towards either of the phase is going to be more attracted towards a carb or directed towards the lipid. And also if that choice is dictated by, as well as I mentioned, is dictated by the nutrient extraction. And how will that relate to the question why is that, of course, even we as humans, we don't do something without a reason. So if you get up in the morning and you eat breakfast, I mean, I only eat breakfast when I know I'm not going to eat lunch. So I'm like, okay, I probably need to be full of energy, and right now I just had a lot of coffee because I was like, yeah, I have to give this presentation. So primarily I hope that is the case for the locust as well, that why they are eating this, why are they specifically in either of the phase, why are they having this specific nutrient choice, and why are they having that? We are connecting that to the migratory studies. So we want to see that what they eat has any relation to do with their migration, such as to the either of the phase goes slow versus fast and long versus short. So if you're in either of the phase, are you gonna fly slower and at a longer distance, or if you're gonna fly very fast but will only go for a shorter distance? And also understanding the anti-predator toxins or such that if that has an effect. So breaking it down to more simpler idea, here we have, you can see on my screen, we have Cysticerca gregaria sartorius phase and the gregarious phase. And we know that the sartorius phase, Cysticerca gregaria are more selective eaters, and the hypothesis is to minimize the nutritional error. So they also eat less on an unbalanced diet. So since they're eating less, we hypothesize that when they migrate and they go under under migration as we impose upon them, they will rely more on the glycogen reserves. And why do we hypothesize that? It's because if they're eating less, then we primarily believe that they will store more of that energy of what they're extracting as glycogen reserves. Whereas in the case of cystosarcoma gregaria, in their gregarious form, they're more nutritionally heterogeneous, so they like to eat whatever they can, and they eat just as much on an unbalanced diet. So as they're eating more and more, we are not saying that the sartorius phase will not have any lipid reserves. We're just saying that maybe the sartorius phase in cystosarcoma gregaria will have higher glycogen reserves, whereas the gregarious phase of cystosarcoma gregaria will have a higher dependency on the lipid reserves because they're eating more, so they can store more of that energy as lipids because they have just so much of it, and there's so much that they can store as glycogen, so abundance of that will be stored as lipids. And such that if that will affect the migration, or if any, such as what would be the flight duration, once both of them we when we impose migration upon them. So how are we going to do that? We have these wind tunnels that we have, so we attach a magnet to the locust over here and we connect them, so they're like connected, and then we blow the air through and they're like forced to fly, and this is how we record how longer they fly and what is the duration of their flight. And these are the lab-based flight experiments at ASU that we hope to be conducting in respect to, again, the trying to understand the phase dependency on the nutritional physiology. So again, since we are just starting, and I'm also a first year PhD student, so we are just starting to reveal what my research interests are and what we're going to be doing in the lab. So that is very preliminary. If anybody has suggestions or questions, it would be a good time to ask.
Thank you. Anyone have questions?
So if I'll ask a quick question, I don't know if people have already studied this or not, but in just the Schistosarca gregaria can fly way longer than the Americana. Is there already known kind of differences in the diet that could be at play there, or are you looking at kind of the differences in across species as well as between the phases?
So we are looking at the differences between the phases. So this experiment is particularly looking at the difference between the phases and what they're relying on as their, or whether glycogen or whether they're relying on lipids. We do know that, yes, the gregaria flies for much longer distances compared to Americana, but we are not comparing Cystosarca gregaria to Americana. We are just comparing within the phase change, and if the phase change affects the nutritional physiology, and if that nutritional physiology is linked to the migration, and if it is, then whether the carb and the lipids dictate, and if they do, then is it the glycogen or the lipid reserves that cause them to fly for longer distance and at faster speed?
Are there any other comments? Okay, if not, I will share the presentation of Alexis. So in meantime, while I'm sharing her presentation, if you have question, please go to the Slido and put your question in there, because I cannot answer any of that because I'm just sharing.
Good afternoon, everyone. My name is Alexis Akoff, and I work in Dr. Brittany Peterson's lab at SIUE, my partner Blessing in the background. And we are investigating how the microbiome influences Schistocerca polyphenism. So basically, we want to see how these grasshoppers can go from one way looking when they're solitary or isolated to being phenotypically different and gregarized and swarming or causing plagues.
Now, did you hear that? Yes? Okay. Okay.
So our major question is, what is causing this phase change of these locusts, and how does this density-dependent plasticity occur? So one of our hypotheses is that rearing condition is a source of intraspecific variation in locust gut microbial community composition. And then another hypothesis that we have is the composition of the gut bacterial communities in Schistocerca species are density dependent. So what we've done in the lab so far has just been some preliminary dissection, DNA extraction, and running some gels. So we've been using some of our older lab-reared colonies, and as well as some older wild-caught colonies just to practice our dissection protocol. With dissection, we remove the crop, foregut, midgut, and hindgut, all intact, and we cryopreserve them in homogenizing tubes until we're ready for DNA extraction. For DNA extraction, we homogenize the guts, run our DNA extraction protocol, and then run a gel just to make sure that we have DNA in our samples. In one of the photos below, we're kind of comparing. At the top is wild-caught guts homogenized, and at the bottom is lab-reared guts that are homogenized. So there's clearly a difference in pigment, and that's something else I'd want to investigate later. So some future work that I hope to do for my thesis in particular is to hopefully visit one of the labs that rears the locusts for us. Once they get here, they're already dead for us to dissect and perform DNA extraction and PCR. So hopefully this summer I can visit one of the labs to rear and treat them antibiotically to alter the microbiome and see how that will play a role in their gregarization. So I want to see if gregarization happens at all, or if it takes longer for gregarization, or if that gregarization looks different, because ultimately if we can alter the gregarization, we can prevent swarming and plagues. So after antibiotic treatment, at the 4th instar, I will perform a behavioral assay test. So just dropping a locust in a box with one side having multiple locusts and the other having none, and just seeing where that locust veers off to, where they spend most of their time, just to see if they prefer to be in a social environment with higher populations of other gregarized locusts, or they prefer to be isolated. So that's just kind of what we've been working on here and some future works. So thank you.
Okay. I hope you hear the presentation from Alexis. So if you have question, please type your question into the Slido. Then I can share with her, and from next meeting we can ask her to or the comments that we get from that. So next is a very quick reminder about the 2 things that we are going to implement in next few months that related to the trainees. First one is we are trying to implement a memorandum of understanding that is kind of show what is the requirements as a trainee you have to do for BPRI and what the mentors will do for you. So this is kind of way to have a transparent communication between trainees and the mentors, and also equal the all mentorship that receiving from the trainees to the mentors. So that is the one thing that we are trying to do in next couple of months. And the next one is in align with that, we will implement the individual development plan. Who do not have that plan, how to develop a new one, and also who have a plan need to change a little bit to add few goals that related to the BPRI. So that's the two things. And the next thing is I wanted to ask you to comment on two questions about when we are good time to meet for December meeting. This is our November meeting. And what is the preferred time for spring semester monthly meeting? So if we can agree on a date, for example, like first week of month, Monday, 1 to 2, kind of that one, then you can all can plan the all other semester activity around that. Then I hope that doing so, we can improve the participation. So use that link that I've provided through the email and share your feedback. In meantime, I also share the screen with you, then you can vote for the meeting time. Okay. Wait. So for December meeting, if you like, please vote for the date. Okay, I think then we will go with the December 20th for the next meeting. And I want you to move forward with the next one. For the spring semester meeting, can you please suggest a time that everybody can commit to that time in each month until the May that we will have the trainee meeting. For example, I given here each month, fourth week of Friday, 4 to 5, same as now, is the time for the meeting. So if you have suggestion, please put it in there. Then I can kind of collect all the comments and decide which date and time is good. Okay, I think I will move into the next one. So I think today meeting, this is end. So how do you feel about the researchers presentation? Please mark your rating, whether it is, oh gosh, it is terrible, definitely terrible, I did a bad from the sharing the presentation. Or if it is going well, we can continue this process. Okay, I think I got the response for everything. Hopefully I will record everything. So thank you very much for your time and coming to this the session and sharing your research. And also before I'm closing this one session, for the all speakers, is it okay for me to have the recording uploaded to YouTube and share as a like personalized link, not as a public access, with every trainees? Is it okay with every presenters? Please at least wave your hand if you're okay, because content belongs to you, not to me. Okay, so I will share with your presentation with everybody, and thank you very much for your time, and hopefully we can meet again on December 20th for the December meeting. I believe that December 20th meeting we will have more participation because no end semester exam. So thank you very much for your time and hope to see you soon on December 20th.
All right, thank you.
Thank you.
Thank you.